Welcome to the deep dive. Today, we're jumping into something really fundamental, genetic fidelity, specifically how our DNA gets copied with just mind-boggling accuracy.
It's an amazing process. And our sources actually start with a great visual. Identical twins, like Mary and Sherry in the text.
Right, we look at them and say, they have the same genes. But technically, that's not quite precise, is it?
Not exactly. It's more accurate to say they contain progeny replicas of the original set of genes from that first fertilized egg.
Protein replicas, okay. And that distinction matters
Because it highlights the sheer scale of replication. You go from one cell to trillions, that DNA blueprint gets copied countless times. And while the process is, extraordinarily accurate, it's not perfect.
So even identical twins aren't absolutely identical at the molecular level because tiny errors creep in over all those cell division.
Exactly. Those minute differences accumulate. But the fact they are so similar really drives home how incredible the copying machinery must be.
Okay, so that's our mission for this deep dive. Unpack that machinery. How fast is it? How accurate?
It's astonishingly fast. We're talking up to 30,000 nucleotides per minute in bacteria. Humans are a bit slower, maybe 3,000 per minute. But the accuracy, it's about 1 mistake per billion nucleotides added.
1 in a billion. That's hard to even conceptualize. Like one typo in, what, a massive library?
Yeah, the source mentions something like 2,000 copies of the Library of Congress with only a single typo. It's phenomenal precision.
Incredible. And the foundational concept, the starting point for understanding all this is...
Semiconservative replication. That was the model Watson and Crick proposed.
Semiconservative, meaning?
Meaning when DNA copies, the original double helix unwinds, and each of those original strands serves as a template for making a new complementary strand.
So each new DNA molecule is half old, half new.
Precisely. Semi-conserved. It keeps half the parental molecule. This was elegant, and it made more sense than the other ideas floating around, like the conservative model where the old helix stayed intact, or the dispersive one where everything got chopped up and mixed.
And the proof, that came from a classic experiment, right? Meselson and Stahl.
Oh, absolutely legendary. 1958, they used E. coli and different isotopes of nitrogen, heavy nitrogen, textolon, and the normal light one in orotext.
How did that work? Isotopes are just heavier versions of the atom.
Right, so they grew bacteria in a medium with only heavy nitrogen. All the DNA the bacteria made incorporated this 15 text, making it dense, heavy DNA.
Okay, so all the DNA is tagged as heavy.
Then they moved these bacteria into a medium with only light nitrogen and let them divide just once.
One round of replication.
Yep. And they extracted the DNA and used cesium chloride density gradient centrifugation. It's a technique that separates molecules based on their density.
And what did they find?
They found one band of DNA. And its density was exactly intermediate between the heavy 15 DNA and the light 14 DNA, a perfect hybrid.
Ah, so not two separate bands, one heavy, one light, which is what you'd expect if the original DNA stayed completely intact, like in the conservative model.
Exactly. That single hybrid band was the smoking gun for the semi-conservative model. Each new DNA molecule had one old heavy strand and one new light strand.
Brilliant. Did they check further generations?
They did. After a second round of replication in the light medium, they got 2 bands, one still at the hybrid density and a new one at the light density.
Which makes perfect sense. Those hybrid molecules replicated again, separating their heavy and light strands and getting new light partners. So you end up with half hybrid, half light.
Precisely. It beautifully confirmed the semi-conservative mechanism.
And this wasn't just some weird bacterial thing.
Nope. Taylor, Woods, and Hughes did similar experiments around the same time using broad bean plants, Physiophaga, and radioactive labeling. They saw the same pattern in eukaryotic chromosomes. It's a universal mechanism.
Okay, so replication is semi-conservative. Where does it actually begin on the DNA molecule?
It starts at specific sites called origins of replication.
Origins, okay.
And the whole chunk of DNA that gets replicated starting from one origin is called a replicon.
So simple organisms versus complex ones.
In bacteria like E. coli, the chromosome is usually small and circular, so it's typically just one single replicon. It has one main origin called oriC.
Oric. And is there anything special about that DNA sequence?
Yeah, it's about 245 base pairs long and notably rich in AT base pairs.
Adenine and thymine, why AT rich?
Because A and T bases are linked by only 2 hydrogen bonds, whereas guanine and cytosine GC have 3.
Fewer bonds means weaker attraction.
Exactly. It takes less energy to pull apart the AT-rich regions. So the cell kind of targets these slightly weaker spots to initiate the unwinding of the double helix. It makes sense, right? Start where it's easiest to open up.
Clever. But eukaryotes have massive linear chromosomes. One origin wouldn't cut it, would it?
Not even close. Eukaryotes solve this by having multiple origins of replication along each chromosome, sometimes thousands of them. This creates many replicons working simultaneously.
So they can copy the entire huge genome relatively quickly?
Right, within the time frame of the S phase of the cell cycle, which is when DNA replication happens in eukaryotes.
Okay, so the DNA starts unwinding at an origin. What does that look like?
It creates a structure called a replication fork, which is basically a Y-shaped region where the parental DNA strands are separated and the new strands are being synthesized.
And these forks move.
Yes, John Cairns actually visualized this in E. coli back in the 60s. He saw the circular chromosome replicating, and it looked like the Greek letter theta because there were two forks moving away from the origin.
Two forks. So it goes in both directions from the origin.
That's right. Replication is typically bidirectional. Two replication forks are established at the origin, and they move in opposite directions, copying the DNA as they go. There was some neat work by Schnoss and Inman using phage lambda DNA and denaturation mapping, basically using heat to melt AT-rich regions as markers to prove this bidirectional movement.
Okay, bidirectional forks moving along the DNA. But wait, if you're unwinding a helix, especially a closed circle like in E. coli, doesn't that create problems ahead of the fork, like twisting?
Oh, absolutely. It's a major topological problem. As you unwind the helix at the fork, the DNA ahead of it gets overwound, creating positive supercoils. Think about pulling part of the strands of a rope that's fixed at the other end. It just gets tighter and tighter.
So the whole thing would seize up.
It would, very quickly. The cell needs a way to relieve that torsional stress. It needs a kind of swivel.
And that's where?
Topoisomerases come in. These are crucial enzymes. Topoisomerase I makes temporary single-strand breaks, or nicks, allowing the DNA to rotate and relax the supercoiling one twist at a time.
Like letting the rope untwist through a small cut.
Exactly. And then there's topoisomerase II, which is even more powerful. It makes temporary double-strand breaks, passes another segment of the DNA helix through the break, and then reseals it. This can remove supercoils two at a time. In bacteria, a key topo-T6 enzyme is called DNA gyrase, and it's essential for replication.
Okay, so topoisomerases act as swivels, preventing tangling. Now let's get to the actual synthesis. There's a fundamental rule, right?
Yes, the absolute bedrock rule. All known DNA polymerases can only add new nucleotides to the free 3' hydroxyl group of a pre-existing strand. They synthesize DNA exclusively in the 5' to 3 direction.
5' to 3' only, always adding to the 3' end.
Always. But the two strands in the double helix are antiparallel. They run in opposite directions. One runs 5' to 3', the other runs 3 to 5'.
So at the replication fork where the DNA is unwound, one template strand is oriented correctly for 5' to 3' synthesis towards the fork.
Right. That's the leading strand. The polymerase can just latch on and synthesize continuously, following right behind the unwinding fork. Smooth sailing.
But the other template strand is running the wrong way relative to the fork's movement.
Exactly. That's the lagging strand.
Yeah.
To synthesize DNA on this template while still obeying the 5' to 3 board rule, the polymerase has to work discontinuously.
How does that work?
It synthesizes short stretches of DNA in the 5' to 3 direction, but it does so moving away from the replication fork, kind of backwards in short bursts. As the fork opens up more template, it starts another short burst.
Ah, so it's making little pieces.
Precisely. These short, discontinuous pieces are the famous Okazaki fragments, named after Reiji Okazaki, who discovered them.
And these fragments, are they the same size everywhere?
Interestingly, no. They're generally longer in prokaryotes, maybe 1,000 to 2,000 nucleotides. In eukaryotes, they're much shorter, typically only 100 to 200 nucleotides long.
Okay, so you have all these fragments on the lagging strand. How do they become a continuous strand?
That's the job of DNA links. After the fragments are made, there are still little nicks or gaps in the sugar phosphate backbone between them. DNA ligase comes along and seals these nicks, creating a continuous, unbroken strand. It uses energy, usually from ATP or NAD, to form that final phosphodiester bond.
Got it. Leading strand continuous, lagging strand in Okazaki fragments, joined by ligase. Now, who are the actual enzyme players doing the synthesizing? The polymerases.
Right. But first, there's another issue. DNA polymerases can only add to an existing 3OH group. They can't start a new chain from scratch on a bare template.
So how does synthesis even begin?
It needs a primer, a short starting sequence. And surprisingly, this primer isn't DNA.
It's not. What is it then?
It's a short stretch of RNA synthesized by an enzyme called DNA primase. This primase lays down maybe 10 to 60 nucleotides of RNA complementary to the template. That provides the crucial 3-OH end that DNA polymerase needs to grab onto and start adding DNA nucleotides.
So every DNA strand or every Okazaki fragment actually starts with a little bit of RNA.
Exactly, which of course has to be removed later.
Okay, we'll get to that. So primer in place. Which DNA polymerase takes over? Let's start with bacteria, like E. coli.
In E. coli, the main replication workhorse, the enzyme that synthesizes most of the DNA on both the leading and lagging strands, is DNA polymerase III, often called the replicase.
Poly-3, is it just a single enzyme?
Oh no, it's a huge, complex machine. A holoenzyme made of many different protein subunits. One of its most critical components is the beta subunit dimer, which forms a ring structure.
A ring. What does that do?
It acts as a sliding clamp. This ring completely encircles the DNA template strand and tethers the core polymerase enzyme to the DNA.
Like a safety harness.
Sort of, yeah. It dramatically increases the enzyme's processivity. That means it allows Pol III to synthesize thousands, even millions, of base pairs without falling off the template. It just slides along.
That makes sense for high-speed replication. Okay, so Pol III does the bulk synthesis. What about that RNA primer?
That's where DNA polymerase site comes in. Pol has a sort of cleanup role. It has a special activity, a 5'3" exonuclease activity that allows it to chew away the RNA primer nucleotides from the 5' end.
Okay, it removes the RNA.
And as it removes the RNA, its other activity, its 5'3" polymerase activity, simultaneously fills the gap with DNA nucleotides.
New function. Remove RNA, replace with DNA.
Clever.
Very efficient. Then DNA ligase comes in to seal the final nick left behind.
Right. Now, back to that incredible accuracy, 1 error per billion.
Yeah.
How is that achieved? Just base pairing alone isn't that good, is it?
No, simple chemistry predicts an error rate closer to 1 in 10,000, maybe 1 in 100,000. The huge boost in fidelity comes from proofreading.
Proofreading, like spell check.
Exactly like spell check. Most DNA polymerases, including Pol III and Pol Y and E. coli, have an intrinsic 3 to 5' exonuclease activity. This is separate from the 5'3" primer removal activity of Pol I.
3 to 5 hexanucleoclase. So it works backwards from the direction of synthesis. Correct. As the polymerase adds a new nucleotide, it checks if it's correctly paired with the template. If it senses a mismatch, an incorrect base, this 3' to 5' exonuclease activity immediately becomes active.
What does it do?
It acts like a molecular backspace or delete key. It clips off the incorrect nucleotide it just added from the 3' end.
Removes the mistake?
Removes the mistake, giving the polymerase a second chance to insert the correct nucleotide before moving on. This proofreading step improves fidelity by about 100 to 1,000-fold.
Wow, so base pairing gets you partway, proofreading gets you much closer, that combination reaches the 1 in a billion level.
Pretty much, yeah. It's a multi-layered quality control system.
So when we picture the replication fork, it's not just one enzyme, it's this whole collection, right?
Absolutely. You've got the helicase unwinding the DNA, primase making RNA primers, SSB proteins keeping strands apart, 2 Pol III cores synthesizing 1 leading, 1 lagging, sliding clamps holding them on, topoisomerase relieving stress. This whole coordinated assembly moving together is called the replisome.
The replisome, a molecular machine. Okay, that's the bacterial picture. How does it compare in eukaryotes? Similar principles, but more complex.
Exactly. The core ideas, semiconservative, origins, forks, 5'3" synthesis, leading lagging strands, primers, proofreading, they're all conserved. But eukaryotes add layers of complexity.
Like the timing.
Yes. Unlike bacteria, which can replicate continuously, eukaryotic DNA replication is tightly restricted to the S phase, the synthesis phase, of the cell cycle.
And we already mentioned the multiple replicons needed for those huge chromosomes.
Right, thousands of origins firing, though not necessarily all at once, it's carefully orchestrated. Another big difference is the polymerase team.
More polymerases involved.
Yes. Instead of one main replicase like Pol III, eukaryotes use at least 3 different DNA polymerases working together at the replication fork.
3. Who does what?
Well, polymerase alpha, Pol-A, works in conjunction with primase. It initiates synthesis. It actually extends the RNA primer with a short stretch of DNA nucleotides, maybe 20 or 30.
So it kicks things off after the primer is made.
Kind of like a starter engine. Then there's a polymerase switch. For the leading strand, polymerase epsilon, Pol-A, takes over from Pol-A and handles the continuous process of synthesis.
Okay, Pol epsilon for the leading strand. What about the lagging strand?
That's handled by polymerase delta, Pol-A. It takes over from Pol-I on the lagging strand and synthesizes the Okazaki fragments.
And do Pol delta and epsilon have sliding clamps too?
They do. The eukaryotic sliding clamp is called PCNA, proliferating cell nuclear antigen. It functions very similarly to the bacterial beta clamp, tethering Pol-A and Pol-A to the DNA for process of synthesis.
PCNA. Got it. What about removing the RNA primers in eukaryotes? Does pol delta or epsilon do that?
No, that's another difference. Primer removal is handled by separate specialized enzymes. Nucleases like ribonuclease H1 and FEN1, flopendonuclease 1.
Hmm.
They specifically excise the RNA primers, then pol usually fills the gap, and ligase seals the nick.
So a more distributed set of tasks compared to pol A in bacteria?
Yet more specialized roles. And there's another huge eukaryotic challenge, nucleosomes.
Right, eukaryotic DNA isn't naked. It's wrapped around histone proteins. How do you replicate DNA that's packaged like that?
It's a fascinating problem. Replication machinery has to somehow get through the nucleosomes and then immediately reassemble them on both daughter DNA molecules right behind the fork.
Does it just knock them off and put them back?
It seems to be more integrated than that. The process is tightly coupled. As the fork passes, nucleosomes are disrupted, but the histones, both old ones from the parent DNA and newly synthesized ones, are quickly redeposited onto the two new DNA helices.
So the new DNA gets packaged right away.
Yes, and evidence suggests it happens via a dispersive mechanism at the protein level, meaning the new nucleosomes are a mix of old and newly synthesized histone proteins.
Are there special proteins helping with this?
Yes, chaperone proteins are key. Things like NAP1 help transport histones, and CAF1, chromatin assembly factor 1, is crucial for delivering histones and assembling them onto the newly replicated DNA. Interestingly, CAF1 actually binds to the pCNA sliding clamp.
Ah, so it physically links DNA replication with chromatin assembly?
Exactly. It ensures packaging keeps pace with copying.
Okay, that covers a lot of the eukaryotic complexity. But there's one more unique problem for eukaryotes, right? Because their chromosomes are linear.
Yes, the infamous end-replication problem, or the telomere problem.
What's the issue?
Think about the lagging strand. At the very end of a linear chromosome, when the final RNA primer on the extreme 5-foot end of the newly synthesized strand is removed.
There's no upstream Okazaki fragment, no 3'-age group for DNA polymerase to extend from to fill that gap.
Precisely. So after primer removal, there's inevitably a small gap, the single-stranded overhang, on the template strand, and the newly synthesized lagging strand is slightly shorter than its template.
And if this happens every time the cell divides?
The chromosome would get progressively shorter with each round of replication. You'd lose genetic information from the ends over time.
That sounds bad. How do cells solve this?
With a remarkable enzyme called telomerase, discovered by Elizabeth Blackburn and Carol Greider, which won them the Nobel Prize.
Telomerase, what does it do?
Telomerase is a special kind of reverse transcriptase. It's an enzyme that contains its own RNA molecule inside it.
RNA inside the enzyme.
Yes. This internal RNA acts as a template. Telomerase recognizes the repetitive DNA sequence found at the ends of chromosomes, the telomeres in humans, its TTG repeats. It specifically binds to the 3-foot overhang of the template strand.
The parental strand that's sticking out.
Right, and using its internal RNA template, telomerase extends that 3-foot parental strand further, adding more repeats. It synthesizes DNA using an RNA template.
So it lengthens the original template strand.
Exactly. By extending the template strand, it provides more room for the regular replication machinery, primase, and polymerase to come back and synthesize the complementary strand, filling in more of the lagging strand end. It doesn't fill the original gap directly, but it prevents the net loss of DNA from the chromosome end over generations.
That's a really clever workaround. Is telomerase active everywhere?
Not usually in most normal human somatic cells, body cells. Its activity is high in germ cells, sperm and egg, stem cells, and unfortunately also in many cancer cells.
Ah, so the lack of telomerase in most body cells contributes to chromosome shortening over time.
Yes, this progressive telomere shortening is thought to act as a kind of cellular clock linked to cell senescence, aging, and limiting the number of times a normal cell can divide. Some premature aging syndromes, like progeria, are linked to telomere dysfunction.
And cancer cells often reactivate telomerase.
Correct. By maintaining their telomere length, cancer cells can often overcome the normal limits on cell division and become essentially immortal, which is a hallmark of cancer.
Wow, what an intricate system from start to finish. So let's recap this deep dive. We started with identical twins.
And saw their incredible similarity as proof of an amazing replication process. We learned it's semiconservative, proven by Meselson and Stahl.
Replication starts at origins, proceeds bidirectionally via replication forks, creating replicons. Cells use topoisomerases to deal with the unwinding stress.
Then the synthesis itself, governed by the 5-foot to 3-foot rule, leading to a continuous leading strand and a discontinuous lagging strand made of Okazaki fragments, later joined by leagues.
All requiring an RNA primer laid down by primase. The main synthesis in bacteria is by pole the third using a sliding clamp, with polar removing primers. Eukaryotes use a team, pole alpha, delta, and epsilon with a pCNA clamp and separate nucleases for primer removal.
Plus, that crucial proofreading activity, 3' to 5 exonuclease, built into the polymerases, boosts fidelity enormously. And eukaryotes also have to replicate through nucleosomes, coupling it with chromatin assembly via proteins like CF1.
And finally, the telomere problem in linear chromosomes, solved by the RNA template-carrying enzyme telomerase, which prevents chromosome shortening and has deep links to aging and cancer.
It's just this incredible multi-component machine, the replisome, working with stunning speed and accuracy. The coordination is just mind-blowing.
Absolutely. And going back to the start, the fact that identical twins are so alike really is a daily testament to how well this whole system works, trillions upon trillions of times in our bodies.
But it also makes you think, doesn't it? Even with 1 in a billion accuracy, over a lifetime of cell divisions, errors do happen.
Right. That tiny imperfection rate is still significant on a large scale. It drives both stability and, ultimately, the variation that fuels evolution, something to definitely keep thinking about.
For sure. There's always more to explore in how life manages its most fundamental processes.